Welcome to BrainMap. This seminar series is co-sponsored by the P41 Funded Center for Mesoscale Mapping, housed in the Martinos Center. It's my great pleasure to introduce our speaker today, Dr. Alexander Lin. Dr. Alexander Lin is the director of the Center for Clinical Spectroscopy in the Department of Radiology at Brigham and Women's Hospital and assistant professor of radiology at Harvard Medical School. Dr. Lin is graduate of the California Institute of the Technology, where he completed his master's degree in bioengineering and his doctoral degree in biochemistry and molecular biophysics. He began his training under Dr. Brian Ross in 1997 and was appointed director of clinical service and senior scientist at the magnetic resonance spectroscopy unit at the Huntington Medical Research Institute. In 2007, Dr. Lin undertook a research fellowship at the National Heart, Lung, and Blood Institute at NIH in Bethesda. Dr. Lin commenced at Brigham and Women's Hospital in 2009, where his research has focused on translating magnetic resonance spectroscopy to the clinic, including strong research focused on traumatic brain injury, recently highlighted on a local CBS program on a concussion and articles in the New York Times, CNN, and other media reports on sports-related head injury. He was awarded the ESMRB Young Investigator Award in 2003 for his work on carbon-13 spectroscopy in Alzheimer's disease, and more recently, Young Mentor Award at Harvard Medical School. Dr. Lin is also an investigator at Psychiatric Neuroimaging Laboratory at BWH, visiting research associate at the Center of Magnetic Resonance Research at the University of Illinois, Chicago, and is affiliated with Massachusetts General Hospital and Boston Children's Hospital. He is also co-founder of BrainSpec, an MRS software startup that won the Google machine learning competition. I would like to remind our audience to address any questions using Q&A box or raise your hand at the end of the talk. Dr. Lin, thank you very much for coming today, and the stage is yours.
Great, thank you very much for having me here, and appreciate the introduction. Real honor to be here at the BrainMap sessions. When I was introduced to it, I started watching some of the videos just to see what other things were covered, and gosh, that was a rabbit hole that it really went down. Great topics you guys cover here, and I'm really honored to be part of that whole process. So today I'll be talking about the chemistry of concussions and looking at using magnetic resonance spectroscopy to better understand mild traumatic brain injury. So just an overview. I'll start just giving a brief overview of what spectroscopy is. I think most of the audience here is going to be aware of it, but for the audience online, I'll dive into a little bit more detail. And then I'll cover sports concussion research that we've done in American football, European football, rugby, ice hockey, and collegiate sports. And then throw in a couple of findings that we've also done in those subsequent studies, also looking at imaging findings as well. And then talk a little bit about brain injury outside of sports, and then finally wrap up with an international effort called Enigma, which I'll get to a bit later. All right, so this is a spectrum. For those that are not familiar with it, this is obtained using a standard MRI scanner, just like we get with neuroimaging, but instead of getting pictures of the brain, we look at chemistry of the brain. And each of these different peaks represents a different chemical, and the height of that peak tells you how much you have of that chemical. And so that's basically how we go about interpreting one of these spectra. Now, it looks very different from your standard imaging, so I'll kind of go in detail about what each of these peaks mean. So the tallest peak you see is N-acetylaspartate. It's a chemical that's found in neurons, transported on axons. And so the concentration of NAA in the spectrum is directly proportional to the number of functioning brain cells you have. And so you can imagine this gives us an incredible insight into the brain, because it allows you to find out, what's going on in terms of neurons. And again, all this is in vivo, non-invasive, using MRI technology. Other metabolites we can look at are glutamate, or glutamine, often abbreviated GLX. These are neurotransmitters in the brain, and that allows us to look at things like neurotransmission. The other peak that we have at 3.0 ppm is creatine. Creatine is part of the ATP phosphocreatine pathway, And so this is a marker of energetics within the brain. So if there's any kind of damage to the brain or a decrease in energetics, we'll see decreases in creatine. The next peak over at 3.2 ppm is choline. Choline is a chemical that's bound to the membranes, and so whenever there is membrane turnover, we'll see increases in choline, and that allows us to look at things like tissue damage. And then finally, over here we have minoxidil at about 3.6 ppm. Minoxidil, like NAA, is a chemical that's found in certain types of cells, and instead of neurons, they're actually found in glia cells. And so we often look at them as a marker for glial function, so that we have increases in minoxidil reflective of increased gliosis, decreases in minoxidil reflective of damage to the glia. It's also polyol sugar. It's also metabolized in a number of other different functions. And that's true of all of these different metabolites. These are all chemicals that are found in metabolic processes all throughout the brain. And so their role in the way we kind of present it will be primarily through this as a neuronal marker, neurotransmitter, energy, membrane, and glia. But it's also involved in a number of other metabolic processes as well. It's something to always keep in mind. And so what we'll do is really try to use these markers as a way of getting a better understanding of what's going on pathophysiologically in the brain. There's some additional ones that aren't listed here because there are peaks that kind of co-resonate with others. But there's glutathione, which is an antioxidant in the brain. So this is the primary way of getting rid of reactive oxidative species. And so any changes in glutathione will reflect that kind of activity. And we also have gamma aminobutyric acid, or GABA, which is an inhibitory neurotransmitter, so sort of the opposite of glutamate. It's an excitatory neurotransmitter. And so this gives us a lot of insight into what's going on in the brain. And there are a number of different ways that we can utilize this within the context of brain injury. And I think one of the reasons why we like using MR spectroscopy, it's fast. To take a single voxel takes about like 2 to 3 minutes, and you get all this information about NAA, creatine, choline, minoxidil with that acquisition. We can look at different parts of the brain, as you see here. And one of the things I think particularly important in the field of brain injury is an objective biomarker. Oftentimes when we talk about concussion, it's often self-report, oh yeah, I got that concussion 2 days ago, or the hit was that hard. I'm okay. I can go back out and play. And so what we really wanted to find was something that was very objective. And the benefit of spectroscopy is it's quantitative. We can look and measure all of these different metabolite levels and get a result back that allows us to really quantify the degree of injury that we're looking at. And so this is why spectroscopy started to become of interest in concussion. Also, the underlying pathophysiology really points to why spectroscopy is important. What we know immediately after concussion, or when the brain is hit, you'll have the calcium grates flood open, potassium channels open. We know from PET experiments that glucose levels change, and we also know that there's appearance of glutamate. But this is on the time scale of really minutes after injury. So the question is, what happens hours after injury or days after injury? What's going on biochemically in the brain? And this is where MR spectroscopy can be potentially helpful. So the first work that I had done was actually severe brain injury. So we're looking at car accidents where the patient has pretty severe amount of injury, and we used MR spectroscopy back at the time when I was at Huntington Medical Research Institutes to look at whether or not it can predict outcome in these patients. So here's a spectrum on top. This is a normal control, just like the spectrum I showed earlier. And then this is a patient that has good outcome. And you can see here that the two spectra aren't all that different, right? For those that are a little bit more experienced, you might see, okay, the NAA to creatine ratio is a little bit reduced. And so that tells us that, as you remember, NAA is a marker of neuronal health. And so we have some damage to the brain and showing a decrease in NAA. But it looks like it's about a 10 to 15% decrease. So you think about it, if you lose about 10 to 50% of your brain cells, not good, but you're probably going to be okay. And those patients obviously reflect a good outcome. In those patients who unfortunately do not have good outcome and we stay in a persistent vegetative state, so that's what PVS stands for, you can see that NAA levels are much, much lower. Now we have about 50% of your brain cells that are now gone. And you start seeing some other changes. For example, you start seeing here at 1.3 ppm, one of the metabolites I did mention is lactate. Lactate is the end product of anaerobic glycolysis. Remember your days of biochemistry. But what that really means is basically, if you see lactate, that means there's lack of oxygen in the brain. And that obviously is pretty devastating. And you can see here in those patients that died, that lactate level becomes very, very apparent. And so using these measures, looking at changes in NAA, decreases in NAA, increases in lactate, as well as an increase in glutamate that we see over here. So you can see here in those patients that died, the elevated glutamate levels, you can actually predict whether or not a patient is going to, how they're going to be after this severe brain injury. We do this on a regular basis at the Brigham, more in post-cardiac arrest, but it uses the same premise. Where looking at the spectrum, this is from a single brain region, 3-minute scan, quantitative results to tell you, is this patient going to make it or not? And so this is really the basis for severe brain injury. So the question, of course, is, all right, we know in a major accident, of course, the brain's going to be impacted. But what about something that's a little bit more mild? What about concussions, as we have seen in sports and people talk about? And I think what was certainly inspiring for me was looking at the first study that was done by Vagnozzi et al. It's a group in Italy that first started looking at sports concussion. This was a really interesting study. So what you see here in black squares are healthy controls. So it's sort of like your normal range of NAA. And then they looked at people with a single concussion, and they found that within days after the concussion, that NAA levels had dropped. And so you can see that it's much lower than the normal range of NAA, again, indicative of some type of brain injury. And then you can see that it slowly recovers back up to about 30 days, back up into that normal range. And what was particularly interesting is they also looked at subjects who had 2 concussions within that same time frame, so a double concussion. And you can see that their NAA levels dropped, that their recovery rate was much, much, much slower. And so that really got us thinking, right? If single concussion, we're starting to see some effects. And then you have 2 concussions. What about like all these concussions that these football players start suffering from? These people have had hundreds of concussions on the field. And what would be the effect of there? And right about the time we started doing this research was about the time that, it became very, there was some unfortunate suicides that resulted from football players. And the idea that there was some long-term changes that are occurring in the brain were starting to become evident. And so our first study that was published, we looked at a small cohort of those subjects. And right away, we saw some big changes in these patients. We noticed that they had increased glutamate levels, which may be reflective of excitotoxicity. And we also saw increased choline. Remember, choline is a membrane marker, and so that most likely reflected diffuse axonal injury that occurred in these patients. And so even with, I think this was a group of like 10 subjects, we were already seeing some big changes in these players. And so this eventually launched into a R1 study that now is a UO1 multi-site center study looking at football players. So we've collected data about over 300 football players. There's also the football players health study here going on at Harvard, which is also looking at the effects of playing football and what it does to the brain and body. So this is the subsequent R1 study findings. So there we scanned about 130 football players and looked in 3 different brain regions: the anterior cingulate, posterior cingulate, and the parietal white matter. Look to see, do we see any changes in our football players? And I think one thing we certainly found right off bat that you can see from the spectra, they're not all that different, right? And that bodes well, right? That for the football players, that their brains aren't totally destroyed, but that there definitely is a range of effects that we see in concussion, right? And I think if you think about it, there are going to be people who are linebackers who get concussed on a regular basis, and even in practice. And the effects of the impact of cumulative effects of impact are going to be greater than compared to, say, a quarterback who certainly gets concussed, but maybe less often than other players. And so there's definitely going to be a range of impact that we're going to see here. And that's what's nice, is that it allows us then to get a breadth of exposure. And so what we see here on the right-hand side, for example, is looking at creatine measures. And you can see how the creatine levels decrease with more and more exposure to head impact. So on the bottom here is the cumulative head impact index. And you can see that as that increases, the amount of creatine decreases. So it tells us that there's some impact on the brain energetics as a result of these concussions. The other thing that allows us to really get into what's going on in terms of the underlying pathophysiology are looking at these markers through the eyes of inflammation. We know that choline, as I mentioned before, is comprised of glycerphosphocholline and phosphocholine. These are constituents of the membrane phospholipids, and that some studies have demonstrated that membrane turnover is associated with neuroinflammation. So if we look at, for example, multiple sclerosis studies, all those patients have very high choline levels, and that's reflective of the neuroinflammatory changes that are occurring in that disease. We also have, and I mentioned earlier, that glutamate is one of the neurotransmitters we can look at. We know that's released by glial activation, and oftentimes what people have argued is that when we see high levels of glutamate, that's an excitotoxic effect, and that's also reflective of inflammatory processes. Myoinositol, I mentioned earlier, is a metabolite that's found in glial cells, and so again, if you have increased glial activation, we all expect to see increases in myoinositol. And of course, glutathione being an antioxidant, that's a direct measure really of what's going on in terms of oxidation in the brain, and allows us to look at that. So in light of that, we looked at glutamate, glutathione, and myoinositol. And so, what were the kind of changes were they associated with within these football players? And what we found was that many of the metabolites correlated with brain and behavioral changes within these patients. And so one of the things that happens with these football players as they age, they start developing symptoms that reflect, kind of neurodegenerative changes. So we have memory loss is one key one, but there's also what's a little bit different from things like Alzheimer's disease is that there's other factors like anxiety and depression that unfortunately ultimately sometimes lead to suicide in some of these players. So this is something that's really important for us to try to quantify in these football players. And what we're finding is that indeed changes in glutamate, glutathione, and myoinositol all reflect these neuroinflammatory effects that are occurring within these football players.
These are 3Ts?
These are all 3T spectra, that's correct, yeah. So this then takes us to the next study that we did, which was then looking at soccer players, or European football, I guess, to be appropriate. This was a great study. I always like telling the story. Dr. Inga Korda is the PI of the study. And when she first started studying this, she went to the soccer teams in Munich and said, we'd love to study concussion. Can we study some of your players? And they said, sure. We're like, wow, that's surprising. Normally FIFA is kind of, very careful about talking about concussion, that kind of thing. And so, of course, when they sent us their players, none of them ever had any concussions in the history of their entire play. And so they were obviously hoping that we would find anything in these players. And so it was really interesting. What we ended up doing was we scanned those soccer players. These are all retired soccer players. And then compared them against a cohort of equally active sports athletes of the same age that have also no history of concussion, but also no history of playing soccer. And so what you see here are some of the differences. In red are each of the soccer players, the spectra, and then in blue is the healthy control data. And you can see that indeed there are some differences, right? We see some changes in NAA, glutamate, choline, and myoinositol. And that tells us that even though these players never had any kind of concussion, we're still seeing some kind of effect in there. And so we started going, oh, what is this effect that we're seeing? And so we thought about it, well, when soccer players play the game, they're always heading the soccer ball, right? And this is coming with a fairly fast rate, with a serious amount of impact of G's on the brain. And of course, it's hitting right straight head in the forehead. And so we asked these players, can you estimate the number of soccer headings you did in a certain game? And then kind of extrapolated out to the amount of headings done per year over a lifetime their career. And indeed, we saw changes in myoinositol that reflect that. So that tells us that that there's probably some kind of neuroinflammatory effect that we're seeing as a result of even heading on soccer ball. We don't see changes in like NAA and some of the other things, so that for example, NAA is actually somewhat higher in these soccer players rather than lower. So we're not seeing any kind of neuronal damage effect. So we're not arguing that heading of a soccer ball is bad, but we are seeing certainly some effects of it. And so I think this even subconcussive injury where spectroscopy is sensitive enough to pick up some of these kind of changes, and that was obviously exciting for that. We also see changes in glutathione. They're not quite as evident as the myoinositol changes, but again, some of the changes we see in brain oxidation. I participated in another study, Andrew Gardner, who's actually here at MGH for a brief time, but he had collected data in Australian rugby players. And there, again, we saw reduced glutathione in the brain. So again, reflective of that inflammatory effect. The one thing that I thought was really interesting is we also found reduced myonostal. And for those people that do spectroscopy, one of the things they know is that when you have reduced myonostal, that's generally a sign of liver damage in the brain. And for anyone who's played rugby, you know these guys are pretty hefty drinkers, and so we think that the reduction in myonostal actually might be a reflection of alcohol abuse that we find in these players who've been playing for a long time. We also found, in terms of looking at just some of the cognitive scores, that also in some of the tests they did where reduced NAA also reflected reduced dexterity. Although I would say between the cohorts of controls and players, we didn't see any reduction in NAA. So this is just reflective of some of the more, again, very sensitive changes that are occurring in these patients. Another study we did was a collegiate ice hockey study. This is data that was collected in Canada with Paul Eklund, where they looked at the corpus callosum and acquired a spectrum from there, which you see here on the left-hand side. And what was great about this study is that it was a longitudinal study. So we looked at the players pre-season and then looked at them post-season and looked to see what kind of changes occurred within the brain metabolites. And what you see is, what we kind of expected, both in males and females. We see that NAA starts out a little bit higher in the beginning of the season, and then towards the end of the season, it comes out a little bit lower. It's not a huge difference, but enough that reflects. There are some players here that did get concussed, and there are some players that didn't. So it's reflected probably of both the subconcussive and concussive effects that we see in this cohort. But what I thought was really interesting was actually the glutamate findings. Here is where we split it up between males and females. In males, we see exactly what's been reported in all the studies that have been looking at football and all those sports, where you see an increase in glutamate, right? This whole excitotoxic type effect. But in women, while it wasn't significantly different, you can see that it's not actually an increase. In point of fact, it seems to be a decrease that's occurring in women. And so that tells us that the way that women get affected by concussions is going to be different from what we see in men. And that was sort of what launched us then into an R01 study to look at what are the differences in males and females in concussion. So we have an ongoing study right now looking at Harvard Collegiate athletes. And so similar type design where we collect the data it's actually a different design where we collect the data immediately after concussion, and then we look to see how subsequently do the metabolites resolve over time. And so, that study is still in works, but we're already starting to see some findings even within the first time point where we measure them immediately after concussion, where we look at myonostal levels and then looking at single task cadence and gait speed. So these are both measures that are often used in the sports medicine world to see what is the cognitive effects. So they'll have someone walk down the hallway and then measure how long it takes for them to walk down the hallway. And then they'll actually ask them to do something else. And I don't know, but something like kind of, you think about the rubbing of the tummy and the head thing, it's sometimes difficult to focus on doing two things at one time. This is the same idea as we do one task while they're walking. How does that affect? That's a really good measure of what's affecting the cognitive measures. And we can see here, there's really nice, strong correlations between myonostal levels and the gait speed. So as the speed increases, we see, it's the way to look at it is, as the gait speed decreases, as it gets slower as we go to the left, we see higher myonostal levels. So again, indicative of that neuroinflammation. Yep. Oh, that's a great question. So the question is, in all the studies, how do we determine the box location? It's not a simple answer to it. We typically look at the posterior cingulate gyrus. And the reason why we did that is because all the earlier severe brain injury studies found that area was particularly sensitive. One of the reasons I like the posterior cingulate is because it gives us the best possible data with spectroscopy. So when we look at these normative ranges, right, it'll be a plus or minus, 2 or 3%. And so if I see like a 10% change in our patient group, I'm going to be sensitive enough to pick that up. Whereas other brain regions, for example, like the temporal lobes, spectroscopy is not going to be as reproducible in that area. And so the variation you have just within your normal population is going to be very high. You're not going to be able to pick up subtle changes. But there is also arguments that, of course, injury might be more prevalent in certain brain regions, and I'll get to that towards the end in the study that looks at things. So it depends a little bit on the sports we're looking at. So, for example, in American football, if you look at the pattern of concussions that occur, and you look at where like you put sensors in the helmets and look to see what are the G-forces that occur. There's a really nice study done at UPenn where it shows it's all over, right? The impacts are coming left, right, front, center. And so in that sense, there is not a lateralization to that aspect of things. But it again depends on certain sports, may show some lateralization. So far we haven't seen that in our data. There are also a number of changes that differ between gray matter and white matter. And so, looking at the white matter also will be also important to try to determine some of the changes that we see. For example, as later on, I'll talk about that correspond with things like diffusion tensor imaging, those kind of things. Yeah, so actually this thing is perfect. Actually, this takes us to the next paper. This is done by Julie Joyce, who's actually here in the audience today and recently joined my lab back when she was with Ashley Harris at the University of Calgary. Did an incredible meta-analysis of all the TBI studies with spectroscopy that have ever been done. So one thing to add, certainly the work that I'm showing here is just representative of the work that I've done. There's a huge body of work. In fact, in this meta-analysis, 118 papers that people have looked at spectroscopy in brain injury. And this gets exactly to the question you're asking, what are the changes throughout the different parts of the brain? So with this meta-analysis, she was able to put together a map, basically, of the brain and what are all the spectral changes. So you can see, for example, in the frontal part of the brain, decreased NAA, decreased glutamate, corpus callosum, decreased NAA, but increased choline, increased glutamate, increased minoxidil. The parietal part of the lobe, decreased NAA, increased choline, parietal occipital, similar findings. And so, for example, in the part that we're in the brain, those are our primary findings as well, the decreased NAA and increased choline. And then looking at areas like the temporal lobes where they found decreases in glutamate. So it definitely is dependent on what part of the brain you're looking at. There are going to be subsequent differences in the kind of metabolites that you might pick up. And this review certainly does a really brilliant job of illustrating that. All right, so double check on time. Yeah, good. So I want to talk a little bit about the fact that MR spectroscopy is not something that operates in a vacuum, right? There are also a number of other imaging techniques that are going to be very valuable to better understand what's going on. And so just want to highlight some of the different modalities that we've also done in conjunction with these MR spectroscopy studies. As you can see here in this image, we can do PET imaging, one can do MRI and get measures of morphology, diffusion tensor imaging, and functional imaging. And all these work in conjunction with spectroscopy to better understand what's going on in the brain. So, for example, this is the neuroimaging protocol that we use across most of our studies. So we do your standard the morphological imaging with MP-RAGE and T2 FLAIR. From that, you can get things like gray matter and white matter volumes. You can look at microhemorrhages with the FLAIR. Multi-shell DTI, where you can get things from like fractional exotropy, which looks like gliosis, and all the different kind of measures of diffusion that occur that are generally thought to reflect other aspects of vasogenic edema, myelin sheath integrity, axonal integrity, and then additional analysis using free water to look at extracellular water. And that then correlates nicely then with some of the neuroinflammatory things that we can measure with spectroscopy. With obviously resting state fMRI, we can look at connectivity. And then I don't need to probably repeat all the different things we can then measure with MR spectroscopy. And so these really tie nicely together. And I think the next step really is for us to start looking at multimodal imaging and what kind of ways we can combine this data together in a way that's synthetic and organic. Starting first with structural MRI, one of the things that we see often in athletes or people who have had concussions is cavum septum pellucidized. So what that is that the cavum septum is this little bit of tissue that's between the horns of the ventricle horns, and you can see that it's actually split within those players that have suffered concussion. Now it's not. You can see here that indeed it's a finding that's not unique to head injury. There are certainly some older patients that have this effect as well. and that there's a lot of NFL players that don't have cavum septum flucidum. So it's just, but it is a significantly different group that have that finding in their MRI. Other things that we can look at are cortical thinning. So this was the work that was also done by Inga Korda, looking at soccer players and seeing that a more accelerated aging that's occurring with repetitive head injuries. And so looking at the age of the subjects, you can see here in controls in blue, that pretty much lays, it's generally flat, although we know there's a slight decrease over time, but that the decrease in soccer players is much, much greater, right? So that basically the brains look older in these soccer players who've had years of exposure to repetitive head injury. We've also looked within the football players at white matter abnormalities. So again, these are the FLAIR images, looking at microvascular changes that are occurring. So you can see in these football players, this is a very, this is one subject that we had that had very significant white matter abnormalities. Not all of them were like that, but you can see that, again, the number of white matter abnormalities correlated then with cumulative head impact index. So more exposure to head injury, the more white matter changes. Other measures looking at limbic volume, for example, was done in that study, looking at how does that correlate with emotion, learning, and memory, and neurocognitive measures. Reduced thalamic volume was also examined. So again, looking at volumetrics and showing findings within them. And so this one actually was an age of first exposure. So how early did you start playing football, and subsequently what were the long-term effects of that? The diffusion tensor imaging data showed the higher fractional anisotropy in the corpus callosum. So you can see in different regional analysis, some of the differences. And again, age of first exposure, looking at when players started playing under the age of 2 versus under age of 12 rather, and over the age of 12, there were some differences in the cohort in this very early study that we did. And now we're taking all this data, we started expanding it out towards in a much larger cohort. So in our multi-site study with over 300 subjects, we'll have more data to look at for this. So as I mentioned, the diagnosed CTE is a multi-center study that's looking at football players. We have the football players health study, our study here at the Harvard University Concussion study. And then there's other studies we're involved in Europe. Inga Korda is running the REP Impact study, which is one of the largest studies looking at soccer players, this in kids, and what are the effects of playing soccer in the brain. So a lot of ongoing sports concussion studies. So hopefully be able to report back later of some additional findings that we'll find with these larger cohorts. So just want to touch a little bit on what other areas outside of sports that concussion matter. And one of the big ones that we first started in was actually in military. And so what we find, one of the big issues with soldiers coming back from the war is that they've been exposed to blast injury. But you can imagine, if an IED goes off, you're exposed to the blast wave, but then you probably also had a friend who might have been killed with an IED blast. You have the post-traumatic stress of that whole event. And the problem is the symptoms of TBI and PTSD are actually very, very similar to one another. And so the question is, how do you treat it, right? With post-traumatic stress, you have cognitive therapies that you basically relive the effects of the event and learn to control your emotions with it. Whereas TBI, that treatment is utterly ineffective. So you don't want someone whose effects are really more TBI laden than to be treated with CPT, cognitive processing therapy, or vice versa, right? We want to be able to find treatments that are more direct for it. And so the question is, can we then use MR spectroscopy to separate out these two? And so we, again, studies underway now, both with the VA here in Boston and the Usarium up in Natick, looking at soldiers and some of the differences we see in different parts of the brain in the different metabolites, choline, glutamate, and minoxidil, in terms of comparing them versus the controls versus TBI versus PTSD versus those with TBI and PTSD. And one of the things that we found early on was that looking at military controls versus civilian controls, there's actually a difference between the two. And so you have to be very careful with your control population to make sure that you're using the appropriate control population when studying these soldiers. Another study that we're just starting up now and has been going on for a little while here at MGH is intimate partner violence. So, when it comes to these football players, a lot of times I talk to them and they're like, you know what, I get paid millions of dollars to play the game. And so if I get cussed, that's just part of the game, right? And, that's obviously totally fine. When you look at the amount of concussions that occur as a result of intimate partner violence, they're horrifying, and that's certainly not something that's a choice of lifestyle. And so, the numbers are well known. 1 in 3 women and 1 in 4 men have experienced severe physical violence from intimate partners in lifetime. You look at some of these papers. There's one study that showed that in one woman who was a prostitute that was exposed to 1,000 concussions over the course of her indenture. And so the effect of concussion is just so much more prevalent in these people, and definitely a bigger issue than outside of just sports. And so I think what's been really valuable is taking what we've learned in sports concussion and trying to apply it to this field. And the question is, is spectroscopy going to be similar to what we see in sports concussion, or will we see something very different? And in particular, in our studies looking at TBI and post-traumatic stress, how does the brain injury coupled with some of the psychological effects of intimate partner violence really impact people? So these are ongoing studies that we hope to be able to find out. So Eve Valera here at MGH has funded to do that study where I'm going to co-investigate with Dr. Eve Vertai here. And I'm also involved in a multi-center study looking at this data being collected across New York, Pennsylvania, and in Utah, and trying to get a better understanding of what's going on with intimate partner violence. All right, last bit, just want to talk about some of the kind of international efforts with this. And you can see we're moving more and more towards multi-site studies. And so the question that, with all spectroscopy studies or imaging studies in general, the more data you can get, the better, right? And so I got introduced to this group called the Enigma, which is Enhancing Neuroimaging Genetics Through Meta-Analysis. It's a group out of USC that really tries to bring together people from all across the world and bring their data sets together so that we can do the kind of analysis with imaging that people do in genetics, right? And initially, the goal of it was to really combine genetics with imaging analysis, but now there's a bigger picture of trying to combine data sets together from an imaging perspective. So just to let people know, we co-founded with Brenda Bartnick, I co-founded the Enigma Spectroscopy Working Group. We meet every first Wednesday at 11 a.m. And so if you're interested, go on the website for Enigma, sign up for those meetings. We've been doing some really interesting work. We have people all across the world that were working with us. When we first started, most of it was focused actually on brain injury. We've now actually expanded beyond brain injury to include other diseases as well. And so this was the first paper that we published out of the group, looking at recommendations for how to go about doing spectroscopy studies in traumatic brain injury, so that we can get some guidelines that we can try to start unifying the kind of data that's coming out of these large center studies. And so the hope is that with that, we'll start seeing some more uniformity. Problem is, the reality is the data is not uniform, particularly in spectroscopy. There's all these different methodologies that one can use. And so the subsequent paper we just published earlier this year was looking at harmonization of spectroscopy data across different scanners, different protocols, different brain regions, and how do you go about doing that? So there's a nice way of really looking at the data both from a perspective and retrospective perspective. So obviously, prospectively, you want to try to get all your parameters as similar as possible, and that makes total sense. That's pretty easy. It's really the retrospective analysis that becomes much more challenging. Obviously, kudos to Julie and her massive meta-analysis to try to combine all this data together. There's meta-analyses. There's also mega-analyses methods. So trying to combine the actual standardization of the analysis of individual participant data, and of course, intelligence, different modeling things like combat. There's different ways that we can try to get all that data together to try to get something that's meaningful. And another aspect of it, making sure that we can share data with everyone and share methodologies. So we have from the ISMRM MR study group, MR spectroscopy study group rather. We started MRS Hub, which is a website that contains all the spectroscopy data, spectroscopy software rather, that everyone can get access to use. We have a forum where you can actually ask questions about how to use this software. So for anyone who's a beginner in spectroscopy, this is a great way of doing it. And our next step is to really start collecting these large data sets through these multi-site type studies to do large scale studies. All right, so in conclusion, hopefully I've left enough time here for questions. Demonstrating that MR spectroscopy is really sensitive to mild and subconcussive brain injury, and that we have these metabolites that are going to be very valuable for better understanding the pathophysiological changes that occur in concussion, looking at neuronal health, diffuse axonal injury, brain energetics, neuroinflammation, all these pathophysiological effects that spectroscopy are sensitive to. This is a technique that's ready and available for anyone to add on to different studies. And so part of my mission here at the BrainMap is to get spectroscopy used more and demonstrate it's not really that difficult to do, right? Single voxel, simple, and you get a lot of information from that. All right, last but not least, definitely want to acknowledge the team that has done all this work. Dr. Catherine Breedlove is actually a sports ATC. We brought her in to really drive this program, and she's been doing that. We have others in the lab, postdocs that have helped process the data, and others that have been involved in the data analysis and collection of the data. I really appreciate all the team members that have done that. And of course, now our new one, sorry, I realize it's not updated to include Julie and Tessa, who are here in the audience, who are now furthering that research. And then, of course, this work that definitely can't be done without our amazing collaborators. Dr. Ross Stefant, over here near the Spalding, has been a wonderful collaborator. Dr. Marty Shenton at the Psychiatric Neuroimaging Lab. Dr. Bob Stern from Boston University, who heads up the Diagnose study. And of course, you heard me mention Dr. Inga Korda. She's over at LMU, but also here at Harvard as well. I've done a lot of work together with her. And David Howell at the University of Denver. Thank you very much, and I'm open to questions.